It's my pleasure and honor really to talk about our institute, about the MRC LMS. And I would like to thank Fiona for giving us the opportunity really to talk to the UKRA community about what we do. There are lots of things that could be said about, could be told about our institute. And we could obviously spend hours talking about details of our science and about what we are excited about. But the specific thing I would like to cover today, or the specific aspect, is really the unique feature of our institute, and that is a biomedical institute located on the medical campus. So that's the feature I would like to cover today, how we marry the fundamental discovery science and the clinical environment, and how that operates in the context of LMS, and how we are dealing with this challenge, which several major biomedical campuses across Europe and across the world are trying to tackle. So before I start, I would like to also thank my colleagues who are also online and who contributed to this presentation by shooting a videos, which you will hear during my presentation. Okay, so I think I better get started because I have lots of slides. So to understand really how LMS operates and how it all came about, we need to go back into history. And I think that's important to mention, to understand the development of this, this marriage between fundamental science and clinical environment. So for this, we need to go back some 80 years or more, where in 1930s, at the beginning of 1930s, at the Hammersmith Hospital, a Royal Postgraduate Medical School was established. So as you can imagine, this was 1930s, at the time of dire economic situation worldwide and also in the UK. And it's really remarkable that the government and the City of London managed to get this funding and get this money together to open the school. The school really had three main missions or tasks when it was open. It was meant to educate the new generation of general practitioners, train specialists, but also to pursue research and into advanced medical knowledge. So you may think this is, from our perspective, modern perspective now, this is not unusual. We all understand that discovery research or preclinical research really is fundamental to medical advances, and it's absolutely necessary to drive the medical advances. At that time in 1930s, that was really quite revolutionary. And really the success of the Royal Postgraduate Medical School is attributed to two key men in the history at the beginning of this, when the school was founded. And the one to mention is really Francis Fraser, who was the founder of this school and was among the first staff appointed. So the revolutionary thing that time was that Francis Fraser was convinced that really the clinical facilities should be run by academics. And there was, especially for medical colleagues of that time, that seemed to be problematic. And he was facing fierce disagreement over this concept. For many practitioners or medics that time, the concept was that the laboratory is needed in medicine only for pathologists, and there is no reason really or no fundamental advantage of having research at the same time or collocated with hospital or with the clinical environment. So that was something which was really truly revolutionary at that time in Hammersmith campus. When then the Second World War started and Francis Fraser was called to deal away from the school to deal with the war response, he recruited John McMichael, who was really the second person who shaped the history of the Royal Postgraduate Medical School. He slightly changed the medical focus of the school, but further strengthened the international research reputation. And I think one of the quotes when I was reading all the historical material stayed in my mind. And that was that his understanding or his motto really was that he believed in great intellectual freedom that in his words, was never allowed to degenerate into a license. And I think that's really interesting thing and very visionary if you think about it in kind of concept of medicine. So after the war, Francis Fraser returned, and the Department of Medicine really enjoyed about 20 years of high fly, so to say. And all the historical material talks about accelerating time with for the School of Medicine, delivering truly revolutionary approach to medicine. No established medical belief would go unchallenged at the time. So truly medicine or hospital run and medical school run by academics at the time. So this was all obviously 1930s to 1950s, when then in 1960s or so, the influence of the school slowly declined. Then at the time, the MRC started to think about how to really address this problem, how to continue this coexistence of marriage or this, of fundamental science or discovery science and medical environment. So to address that in 1970s, Clinical Research Centre was formed in Norfolk Park in Harrow in North London that existed for some years, but didn't really work that well as the original Royal Postgraduate Medical School. And then eventually in 1994, the MRC decided to go back to the Hammersmith campus and to establish the MRC Clinical Sciences Center, as it was called at that time, to build on this experience on the campus and on the historical perspective and success of the Royal Postgraduate Medical School. The mission of the MRC CSC, as it was called at that time, was to undertake high quality basic science, which would underpin our understanding of the human disease, because it was located in this context of the Hammersmith Hospital campus, and also to facilitate the translation of this understanding to improve diagnosis and treatment. So then in 1998, so this was 1994, 4 years later, the CSC became the third directly core-funded MRC institute. So that time next to LMB and NIMR in Mill Hill. And that was also the time where the partnership with Imperial College got forged, as the MRC CSC, as it was called at that time, became part of the Imperial's Institute of Clinical Sciences, which is one of the departments of the Faculty of Medicine at Imperial College. So one last thing I should say about the history, which is important, is that in 2017, the CSC was finally renamed to the MRC's London Institute of Medical Sciences, really to reflect the institute the status of the MRC core-funded institute. So this is all obviously historical perspective, which I think is important to understand when you think about our institute and how we operate. So LMS these days, of course, Hammersmith Hospital and campus has evolved. And the LMS today is a part of multi-billion investment White City medical and innovation campus. So, which consists of the Hammersmith Hospital campus where LMS is located, and with the hospital complex, but the neighboring White City Innovation District, which houses a number of startup companies and incubators and scale space and invention rooms, lots of new biotech, as well as the White City campus of Imperial College, which where the chemistry got co-located, bioengineering, and School of Public Health. So where LMS sits at the moment is, having a hospital environment on one side, and then having this biotech innovation district on the other side with bioengineering, chemistry, and School of Public Health. So we just form together all this really vibrant and exciting new ecosystem. So the mission of the LMS got slightly rephrased as well over the time. So the current mission builds obviously on the historical mission of the CSC that time. So we are tasked from the MRC to be at the forefront of innovative biomedical research. We obviously, our mission is also to promote translation, as we are located on this hospital campus for maximum medical benefit, but also to work on this within our long-term partnership with Imperial College. We are tasked with training the next generation of leading clinical and non-clinical scientists. That's, again, lots of that is related to our physical co-localization with the hospital. And then finally, one of our missions is to enhance the public interest and in science and do lots of outreach activities. So that's our current mission as an institute. Thinking about our science, we have about 30 groups, core-funded groups in the institute. The research is underpinned by state-of-the-art research facilities. And the research groups are organized at the moment into two distinct sections based on the themes we covered. So the sections are epigenetics and genes and metabolism. And if you think about what we do in those sections, so within the epigenetic section, we deal with or address genome regulation, cell fate, inheritance across cell cycle and across generations, and how environment influences the genome function, the activity of genes. Whereas in the genes and metabolism section, we address, or the groups address, cell signaling, interorgan communication, and whole animal physiology. We do have two areas of science which are cross-cutting across the whole institute, across both of those sections. And that really is the quantitative biology approaches, data science, and artificial intelligence as an approach to understand complex systems and complex data sets. And then our distinct core competence or feature of the institute is also this imaging across scales, where we have the capability and expertise to go from single molecule imaging and volume electron microscopy up to the PET scanning and MRI scanning of patients. So that is really something quite unique to this institute, that we really have the span of capacity to go from single molecule imaging up to the patients. So you may think, why epigenetics and whole animal physiology? Why those two areas, and how that makes sense in this kind of clinical campus environment? And the way we think about it is that, of course, we all inherit our DNA, which from the genome from our parents, but it's really what determines our susceptibility to disease, or how the genome will be translated into phenotype or into physical features, is how this DNA interacts with environment. And part of this interface that's where epigenetics and metabolism and physiology sits. So we really are interested in this interface between our DNA and the environmental factors related to our lifestyles and infection pathologies, and also aging, how this inputs into this kind of interface between formed by epigenetics and metabolism and physiology. And we believe that fundamental understanding of the core principles of that will make us understand why some people are more susceptible to some disease, or some people develop some problems, or some people react in a particular way to treatment while others don't. One of our core missions, as I mentioned, is really training of clinicians in fundamental research. And in this case, we really need to talk about our chain flora scheme. So this is a training scheme which was established back in 2009 by our former director, Professor Dem Amanda Fisher. And that's a scheme which is co-funded by the LMS through the MRC, and by our partners at Imperial College through the National Institute for Health Research and IHR and BRC. So it's a joint scheme co-funded by MRC through us and by this BRC and IHR through Imperial. So the unique thing of this scheme is that it's really designed for clinicians at various stages of their career and to train them in fundamental science. The uniqueness of this scheme is that it's very flexible. So we have various entry points for clinicians at different time points of their training. We have the Foundation Year 2 scheme, Chain Florey Fellow. We have then Research Fellow, which is Chain Florey Research Fellow, which is designed for clinicians to obtain their PhD. And then we have Clinical Lecturer and Senior Clinical Lecturer. And the flexibility of this scheme really allows us to tailor the support for individual clinicians for their needs and for the support they need at the different stage of their career to allow them really to get trained and to operate at this interface between preclinical research and the needs of their clinical training or where they are in their clinical career. So this is the basic overview where we are. And I was thinking the best way really to deal with this talk was not just for me to talk 25 minutes or so, but to hear some case studies or testimonies from my colleagues who are also present, so they will be able to answer your questions at the end. So I hope the sound will be okay in the video. And let me play the video.
So I came to the LMS in 2012, having been in a chemistry department in the US for 7 years. My background is in chemical engineering and physical chemistry, but I've been doing biophysics since my postdoc. I came to the LMS for several reasons. I was looking for an environment where I could develop and take advantage of the tools that I had been developing for the last few years and apply them to more physiological systems and more interesting biological questions. So, for example, a recent work with the Aragon Lab has helped us really team up a number of postdocs and students to tease out the mechanism of an important family of enzymes called the structural maintenance of chromosome complexes, which are involved in the formation of chromosomes, in the segregation of chromosomes, in DNA repair and DNA replication. These are really fundamental protein complexes that structure the chromatin inside the nucleus of eukaryotic cells and regulate gene expression and are really important for the correct division of the cell. And so thanks to the fact that we're right here next to the Aragon lab and Enrique Martinez lab, we now have access to a number of people who are working in both labs, purifying high quality complexes, high purity complexes, and using them on our single molecule machines to really investigate how these DNA loops are made, the fundamental steps of the loops, what factors block them in chromatin, so like nucleosomes, and what chromatin factors such as FACT help these motors form these chromatin loops in cells. Having been trained as a chemical engineer and a physical chemist, I knew very little about cellular biology, and coming here has allowed me to push this RNA Mango technology to develop single molecule imaging tools in mammalian cells at the single molecule level. So we can now track single RNA molecules with very high contrast using these tools. We can look also at small non-coding RNAs or long non-coding RNAs, both in fixed and live cells. And this has been thanks to the help and the support from many groups in the Institute, notably the group of Matthias Merkenschlager, with whom we work very closely as well. And so, so pushing this technology that we were initially developed in chemistry departments and applying them in a more biological setting and physiological setting has helped us, learn a lot about how to use this in useful cellular systems, which is ultimately the physiological environment where these biochemical reactions take place. The benefits of being where we're located has transpired during the last pandemic in 2020. So during the pandemic, we started thinking about how we could help, and it occurred to us that we could use our Mango technology and translate it into a rapid technology for detection of viral RNA. And this has been possible because we're at the interface between Imperial and the LMS, and we're located at Hammersmith Hospital, where we've found the inspiration to develop this new technology that allows us to detect viral RNAs in untreated saliva samples in less than 30 minutes, which is now a technology that we're trying to translate into the clinic and commercialize through a startup company that we recently started.
I have been with my lab at LMS for almost 15 years, and what we have been doing is try to identify genes and pathways that are involved in controlling different aspects of senescence. And this has been funded by MRC during all this time. And what has happened more recently in the last 5 years or so is that we and others have realized that senescent cells are involved in disease, and all the basic knowledge that we have accumulated through trying to study this basic process of senescence during all these years can now be used to try to target senescent cells, kill them specifically, and this can be useful for treating cancer, treating fibrosis, and many different diseases of aging. Being at the LMS, I have been surrounded by different groups that have expertise in other cell biology aspects that I'm very interested. We are also surrounded by clinicians that are working with different diseases, in which now I'm trying to see whether we could target senescence, and this goes from liver disease to cancer to many others. And more recently, being here with all the YCT developments and having all the imperial chemistry, bioengineering has opened the possibility to many different collaborations that go beyond what I can do in my lab and my specific expertise. We have already been more involved in trying to apply some of the basic research that we have into identifying targets and drugs that we can use to treat senescence-associated disease. I think in the next years we will expand into our basic science of trying to understand senescence, but also we'll collaborate with colleagues at chemistry, at YCT, and in the campus clinicians interested in trying to apply and understand the role of senescence in disease.
So I first came to the LMS in 2015, having completed a MB PhD at another big UK institution. I started on what was called a Chain Florey Foundation program, and then was well mentored at that time by Petra Hajkova, who I previously collaborated with. And subsequently I went on to an academic clinical fellowship in pediatrics, which again was funded by the LMS. And during that time, I got my first grant and was supported to independence. And that allowed me to establish my own lab at quite an early stage of clinical training. My lab studies how pluripotent cells in the embryo give rise to the germline, the precursors of sperm and egg. And we study this both in mice, but also in a culture dish using stem cell models from mice and now increasingly humans. And subsequently, I've gone on to specialize in clinical genetics, and I'm now a Chain Florey lecturer, combining my genetics training with running my lab. The LMS has been very supportive of the moves I've made. The realization and support available from the LMS to support clinician scientists. They see our value and they really want to integrate us and work with us in a way that is different to other places and is based on years of experience both within the LMS but also I think more broadly on the Hammersmith campus.
So for many years we've been investigating how the brain and the gut communicate. So you may not know this, but actually our brain and our guts communicate. They're engaging in this bidirectional crosstalk. And this crosstalk is very important for various things. It's important because it regulates how much we eat, how fat we get, and even our mood, right? Whether we're happy or not. So we're trying to understand what kind of signals they exchange and why they do it. We've been doing our research using fruit flies, so the fruit fly Drosophila. So you may think this is an unusual entry point into understanding how our brain and our gut communicate, but it turned out to be a really useful one. And the reason why it's useful is because we can turn many genes or many cells on and off and ask what happens physiologically. So we know that there's a lot of redundancy in our physiology. It's quite hard to uncouple cause from effect in everything that goes on within our bodies. So the flies allow us to actually establish that kind of directionality. So I turn a certain gene off in a particular cell type, and then I see what happens. And the advantage is with flies, we can do that hundreds of genes at a time, if not thousands. So what we discovered using flies somewhat unexpectedly is that our guts have a sex. And what I mean by that is that they know that they're male, whether they're male or female. And you might wonder, well, why does our gut need to be male or female, and does that matter? And what we've also found with flies is that this matters for a number of physiological, so aspects of our physiology, but also our pathophysiology, or actually rather than that of the flies. So what we found is that the gut needs to be male or female in a way that controls how much the fly eats and whether the fly is able to reproduce. So for successful reproduction, the female gut needs to be female and the male gut needs to be male. One question that we're very intrigued by is whether these sex differences that we found in flies are also found in mammalian gastrointestinal tracts and even within our own gastrointestinal tract, right? So does our gut know whether it's male or female? A lot of what we do is very blue skies, and we sort of like to pursue research avenues that are novel and have not been investigated, but this takes time, right? And many of our projects take up to 7 years to be delivered, and I think the LMS provided us with the kind of continued support that we need for this kind of research.
So I lead the computational cardiac imaging group at the LMS, and we use a number of computer vision approaches to analyse cardiac MR imaging mainly in large populations, both in health and disease. We analyse these images to build 3-dimensional models of the heart so we can better understand the structure and function of the heart. And then we link that to genetic information and outcomes to look at potential new causes of heart failure and the genetic risk that is important in developing heart disease. One thing that we are really focused on is moving towards translation. So we have a number of tools that we've prototyped and developed that appear to be very powerful for stratifying patients at different levels of risk. We're working also with NIHR, NHSX to develop software tools that can actually be rolled out into the NHS that could be used in real time to guide patients to the most appropriate therapy and avoid overtreatment as well. We're also working with pharma as well, using data from UK Biobank to identify potential new pathways for treating specific types of heart disease that are related to aging. So we think as well this translational pathway for therapeutics could be really exciting and address an important unmet need, particularly in an aging population and in a population with increased cardiovascular risk factors such as obesity, smoking and diabetes. So for me as a clinician working in the LMS, the real strength is having that co-location between really innovative human imaging on a hospital campus, but also having really strong links with mathematics, with engineering, with cell biology and with genomics. So bringing those two things together on a clinical campus I think is a really unique advantage of working within the LMS that isn't available at other institutions in the UK. One really exciting thing about the work that we're involved with is we move very freely between patient populations, between animal models, and then back again to large populations and particularly collaborating with pharma, accelerating progress towards therapy. So it's really the integration of these very vibrant different research possibilities and the co-location, I think, on a clinical campus that is really pivotal in supporting our research.
So I think those 5 little stories about covering some of the science that is taking place in the LMS, I think demonstrate the key points about the LMS, which are the key strengths. I think those will be the usage of the state-of-the-art technology and innovation to allow and underpin research across different scales and modalities. And it's moving between model organisms to patients to humans, back to model organisms, and particularly going across scales, thinking about different approaches in imaging, as I mentioned previously. I did mention the model organisms because we do have a distinct expertise in use of model organisms that enables us to probe causality, because of course experiment, that's something difficult to address when you just deal with patient samples. I think one of the key strengths and competencies of the LMS is really the interdisciplinary character, and that was demonstrated by David Rueda talking about his physics background and single molecular imaging, and also by this co-localization with the White City campus bioengineers and material scientists and chemists being on the campus, which allows us to interact with them really directly, being in close proximity. And I think of course the unique, the USP of the LMS is really the integration of scientists and clinicians, which allows for the flow of talent and flow of expertise and ideas between fundamental discovery science and the clinical environment. So one of the very few slides I have just to summarize that, you think in the relatively short history of LMS, has this delivered in some way, or are there some examples beyond of success? We all know that science is a long-term undertaking and requires long-term investment to drive innovation and to drive discovery. But even in this relatively short history of the LMS or CSC previously, I think there are some really interesting examples how the science transpired into some clinical outcomes. So some of those would be, this is by no means exhaustive, and this is thinking about the medical outcomes, not about the discoveries in the area of fundamental science. So next to what was mentioned in those little videos, I think what is worth mentioning is the development of the cool cap, which is the cooling of the neonates, brains of the neonates who suffered lack of oxygen during birth. That's now being used in the clinic. Development of candidate treatment for sepsis. Lots of novel imaging methods used for neurological and psychiatric patients. Machine learning models to predict risk of heart failure. And last but not least, there is a simple screening assay has been developed and implemented and commercialized by Illumina for screening 17 different cardiac conditions, and that's based on the research that has that took place at the LMS. So this is where we are, and you will think like where we are going as an institute. And one exciting thing I would really like to mention is, of course, we are getting a new home. At the moment, the LMS, 30 groups are spread physically between 5 different, although interlinked, but still 5 different buildings. And I think it's really exciting that finally we can, as an institute, move under one roof and be all together in one new building. And this is the building which is being finalized at the moment. We are supposed to move next summer in August 2022. So as you can imagine, there is lots of activities and planning happening at the moment. So that's my presentation. Thank you to all of you for joining.